or duplicate, which is a important of synonymous words. But in the definition, the mitosis is a type of cell division in which chromosomes are equally distributed. The chromosomes are equally distributed to the one cell, to the parental cell, to the daughter cells. So in which chromosomes are equally distributed, resulting in two genetically identical daughter cells. One parental cells which can distributed or divides to genetically two similar cells of daughter cells. That is called a mitosis. History mitosis was first discovered in plant cells by the Strasburger 1875. Later on, W. Fleming 1879. 1879 discovered it in animal cells. The term mitosis was coined by the Fleming 1882 occurs. The cells undergoes mitosis are called the mitocytes. In plants, the mitosis are stem cells, generally germinal, epithelial, and embryonic cells. It also occurs during the regeneration. The root tip is the best material to study of mitosis. Root tip, shoot tip, node, internodal, that four major parts, which is active of cell division. That cells are called meristematic cells, which can performing the only growth and development of the cell, also called organisms. Which, so this duration, it varies from 30 minutes to the 3 hours. One cell divisions, 30 minutes, 3 hours. Steps of mitosis. Mitosis is a continuous process, and for better understanding, the whole process is divided into the following stages. First one is the prophase, prometaphase, metaphase, anaphase, telophase, C phase, or cytokinesis. So this is a total 6 stages. Out of the 6 stages, above 5 stages is called a karyokinesis. Last C stage is called a cytokinesis. Karyokinesis and cytokinesis most are responsible to the cell division of mitosis. Mitosis. So this is also mitosis are first one prophase, second one prometaphase, third one metaphase, fourth one anaphase, fifth one telophase, sixth one cytokinesis, seventh one two-cells formation. That is also 7 stages are there in the mitosis. Then first you get the prophase. What are the means of pro? Pro means first. The first form, the nucleus becomes a spherical and cytoplasm becomes more viscous. That is the first one. The nucleus are in a spherical and also it become a nucleus or a spherical in nature. Then the chromatin slowly condenses into the well-defined chromosomes. Then the chromatin body means are a thick part which are slowly condensed to definite chromosome structure. Each chromosome appears as a two sister chromatids joined at the centromere. Two sister chromatids are joined to the center part of the chromosomes, which is also first stages. The spindle or microtubules begins to form an outside nucleus. In plants, the spindle appears or mitochondrial spindle is a anastral. Spindle means disappears in animals and brown algae. The mitosis spindle is a amphistatile. Then anastatil and amphistatile, which is a includes the two esters in opposite poles of the spindle. Each ester consists of two centrioles surrounded by a astral rays, astral and also called non-astral areas, which also major important of the prophase part. This is also prophase. So first point in the interphase after then prophase. Prophase is the chromosome is also nucleus. Nuclear membrane is also disappears. First point. The nucleus in the nucleus condensed bodies changes into the elongated linear model of the chromosomes, which is also prometaphase. The nuclear envelope breaks down into the membrane vesicles and the chromosomes are set free into the cytoplasm. Then the chromosomes are attached a spindle microtubules through kinetochores, specialized protein complex that mature on each centromers are called the kinetochore. The kinetochore is a special part of the during the cell division, which is also that complex that mature each centromeres are called the kinetochores. Then the third one, nucleolus is also disappears. The nucleolus is a disappears, which is also second stages of the mitosis, then prometasis, but is also called the stage of the prometasis. Then here is the interface, protophase, protophase, prometaphase, metaphase, anaphase, telophase. This is also called how many stages are six stages which involvement of the cell division of mitosis. But here is the third one. First one is the prophase, prometaphase, and metaphase. So this metaphase is a kinetochore microtubules align the chromosomes in one plane. So this is also one plane kind of course. This is also one plane manner. Here is also several microtubules are arranged in a pole, south pole to north pole. Between part is called meta phase. In one plane to form the metaphasic, the plate are equatorial plate. The process of the formation of the metaphase plate is called the congregation. The metaphasic part is called a congregation part. So which is important. Then the second centromeres lies on the equatorial plane, while the chromosomes or arms are directed away from the equator called the auto-orientation programs. So this auto-orientation part which occurs in the metaphase, meta means middle part. The chromosomes are arranged in a spindle shape of metaphase or chromosome arrangement. The smaller chromosomes are remain towards the center while larger ones are occupies the periphery. The two chromatids are smaller is also center part and larger chromatids are which can part outside or peripheral part of the chromosomes or nucleus. This is also one type of the part. Then anaphase. Fourth one is anaphase. The chromosomes are split. The chromosomes are split simultaneously at the centromeres, so that the sister chromatids are separate. They are now called the daughter chromosome. So then the chromosomes are which elongated and tubular linear model that is called the daughter chromosomes. The separate sister chromatids move towards opposite poles at the speed 1 millimicron per minute. 1 millimicron per minute. The central chromosomes are which can passes to the towards the opposite poles of the nucleus, opposite poles of the cell. The separate sister chromatids move towards the poleward movement of daughter chromosomes occurs due to the shortening of the kinetochore. Microtubules are appearance and elongation of interjonal fibers. Interjonal fibers. This is also important. The fibers means the hair-like structures, which is also on the hair-like structure. The chromosomes are attached to freely swims in a cytoplasm. Then daughter chromosomes are appears in a V-shaped. V-shaped means metacentric chromosomes. V-shaped structures. So then the metacentric or L phase, submetacentric or S shape, which is also acrocentric, and one as one shaped telocentric. Then it is the shortest of the all stages of the mitosis. Any stages which is a common type. Then also called a fifth one telophase. What is the means of the telo? Telo means tail. Tail means last. Then last one, the daughter chromosomes are arrived at the pole. At the pole, then kinetochore microtubules are disappears. The kinetochore means central part of the chromosome mainly. It is also called a microtubules. And so then it can consist of a kinetochores are also there. The nuclear envelope performs the around each chromosome clusters of the each chromosomes are made up of the clusters. The chromosomes are uncoiled into the chromatin. Uncoiled. The nucleolus are reappear. The first stage lo my prophase lo nucleus are disappears, which are reappears in fifth stage, also called telophase. So this is the nucleus reappears. It is considered as the reverse of the prophase. Reverse of the prophase. This is a important of the character features of the telophase. Then also called as C phase associative genesis. First one, prophase, prometaphase, metaphase, anaphase, telophase. Then six C phase or cytokinesis. C phase, cytokinesis means C phase means then only rest of the cytokinesis which are separate to the two parts. Then the total nucleus divides in two is called a four stages. That is called a prophase, metaphase, anaphase, and telophase. So the C phase or cytokinesis is the cytoplasm individually that starts during the anaphase and completed at the end of the telophase. After complete of the anaphase, it can start of the C stage or cytokinesis stage. It is balanced by two bare different methods, cell plate methods and cleavage or cell furrowing methods. Cleavage also cell covering methods. That is also C stage is also cytokinesis formation. Cytokinesis means the protoplasm like or liquid like organism that can separate the two parts. The cell plate cytokinesis occurs in plate cells. The spindle fibers are persistent and equatorial planes. So the Golgi vesicle fused at the center to form a barrel-shaped formoplast. Further addition of the vesicles, it can causes the formoplast of growing or centrifugationally till it meets with the plasma membrane of the mother cells. This is also important of the reactions which can be separate of the pro-metaphase and metaphase of. After pro-metaphase, metaphase, anaphase, telophase, C phase means C means cytokinesis stage. Cytokinesis means only liquid of protoplast, it may be divided. The content of the formoplast ability to become a cell plate or a future middle lamellae, which separate the two daughter cells. So the daughter importance or the daughter protoplast species are primarily well in the middle as on the both sides of the cell plate are a middle lamellae. This is also only cell plate and aquatic plate. Then this is also last point telophase. Telo means last part of the plant. It is also daughter chromosomes are arrived at the pole. Then the middle part of the chromosomes shifted to the poles. That is also called telophase. So in the kinetochore and microtubules are disappears. Nuclear envelope refers around each chromosomes clusters of each pole. Each chromosomes are clusters for formation of the nuclear envelopes. The chromosomes are uncoiled into the chromatin. Coiled. Uncoiled. Uncoiled means not coiled. Nucleolus reappears. In the first stage, the first stage is also nucleus or nuclear membrane is also disappears, but here is also reappears. The nucleus is also reappears. So this type of the reappears. The third one, nuclear envelope reforms around each chromosome, clusters of each cell, each plate. The chromosomes are uncoiled into the chromatin. The chromosomes are uncoiled. Uncoiled means it is not separated, but here separate, it can be coiled. The chromosomes are uncoiled into the formation. Then the nucleus reappears. Nucleus reappears. It is the considered as the reverse of the prophase. Reverse of the prophase. That is called the constructions. Then C phase. The C phase is a important of the C phase. So the C phase here is sorry. So this is also the C phase. The C phase is cytokinesis phase. Here is a that is a C phase, but here is a cleavage cytokinesis. The cleavage cytokinesis it occurs in animal cells and pollen mother cells. So of a same age, same some angiosomes in this process, a cleavage furrow appears at the middle, which gradually deepens and breaks the parental the parent cell into the two daughter cells. A specific uh, structure called the mid body are formed in the center and is a centromer pitch or centromer primary. This is also important of the cleavage parts. Then what is the differentiation of the karyokinesis and cytokinesis? Then karyo means also cell division of the nucleus. Cytokinesis is also called a protoplasm of the cytoplasm.